I think it is good for us to I think of this as like housekeeping. Every once in a while, we're gonna go back to the syllabus. We can see where all the points in the class lay, what we've done, what we've got to do, right? Lecture exams, we've done through 3, we're in good shape. We've got one more coming up that will be about the fishes and chordates and amphibians, I think. The last lecture exam, the 5th one, is basically about birds and mammals, and there's a little bit of cumulative stuff in terms of cladogram. Okay.
Now, we just take a third one. Or could we just take a fourth?
So we've taken 3. We've got one more coming up about fishes and amphibians. And then the 5th one is like our final. But the majority of that 5th test is just on birds and mammals. Okay. There's a little bit of cumulative stuff which I'll talk about, but it's basically that cladogram on animals. Okay. So that's not going to be anything really huge and new. Okay. The quizzes, of course, those have just been plugging along. Boblet's participation, I'll talk about this. Though in the news, we've already talked about the resume is done. We're done with one of the 2 lab exams. The second lab exam, let's face it, it's going to be easier for you than the first one. Right. It's going to be a lot of the same organs over and over on different species. Right. A lot of this stuff is more familiar with what's a gallbladder, what is a liver, compared to, you know, where does the testes on a Clonorchis or something. That's on the syllabus, you know, here in the schedule. The lab report and the tardigrade pictures. Okay, so let me address the boblets participation. The boblets, again, the date has been fixed for a while now, but what you'll be on the hook for is there's going to be 3 blocks. There's a block on Friday night, and I think what we're going to do there is probably a lot of frog calls, owl calls, maybe some salamander hunting in the creeks. That's 6 to 10. Some of us will probably be camping out. I'm not exactly sure. We might have a bonfire. Saturday is going to be birding, and then probably spider hunting, and then snakes and things. And then Friday or Saturday afternoon is largely going to be fishes and mussels in the creek. Okay. And then there'll be a show and subs at the end. Okay. So you can come Friday night and Saturday morning. You can come Friday night, Saturday evening, or just come on Saturday. And will be those points. Okay. And what you need to do while you're there, I mean, honestly, this is embarrassing to say, but there have been times I just say, you know what, people, just go sit over there in the shade and shut up, you know? That's the really all you have to do is occupy 3-dimensional space and not whine and just stay out of my way. But I'm telling you, that's a really cynical view of this. It is a truly cynical view because this is going to be fun, right? I don't know how many of our students in the past have just said, like, I had never done anything like this before. Getting in the creek, doing seine netting, stopping around, catching fish, bird watching in the morning, holding a snake for the first time ever.
Wait, which one are you holding the snake?
You know, the spider thing I'm really excited about. So I don't think this should not be considered. Honestly, if this sounds like terrible work to you, then you may want to consider, you know, English major or something like that. It's going to be fun. And I know you guys all have great attitude. It's going to be a good time. So that's where those points come from, those boblets. The tardigrade pictures. Okay. Let me show you where that is right now on Canvas. Oh, there are questions? I'm sorry. There are questions about the boblets there? Yeah, read.
So most of our football games are on Saturdays.
Okay, well, is it predictable whether there's gonna be one?
No, because we have, if we make it the playoffs, which we have, it will be then. So if we have a game, for example, if we make it all the way to the finals, or like, to the final four, we will be playing then.
Okay. On Friday and Saturday?
Yes. We will have to be at football Friday. We will either be traveling to whatever location it is on Saturday, or we will be having meetings and practice, which are usually from 5 to 10 at night.
Okay. Well, we'll figure something out,
Okay.
as usual. It comes down to the academics making arrangements for the athletics, and this will be no different either, so we'll figure it out. Because you guys aren't gonna get punished, obviously, by me. You know what I mean?
Yeah, well.
But it's just
others
us,
said-
it's always us that have to be flexible, do extra work, And you can send that to your coaches, because I'm over it,
I understand. He's
But,
been
I
very
hope you don't hear
aggressive
me,
lately, so he's
Yeah.
got.
I hope you don't hear me being like, that you guys, it's anything to do with you. It's mommy and daddy are fighting.
Yeah.
It's not their kids' fault, right? So we'll figure that out. Okay, so
I'm just letting you know that, like. he's
No, I appreciate that.
newborns, so he's gonna be very not lenient. He's very
Yeah.
aggressive
No.
lately, so if you're trying to work out a deal, it might not work out very well. Just being honest with how he's been
I don't
lately.
even bother to work out a deal. I just bend over backwards, kiss my own butt, right?
Yeah, we all have to do that with him.
Oh, no, you guys are caught right in the middle of it. So let's see, the target, are there other questions about I'm really excited about this, the boblets thing. The other thing is you get to interact with other people besides just me, right? So I think that's cool to see other people come on campus that are excited about.
So
About
you
you.
attend 2 out of the 3, or do you just attend 1?
Yeah, so 2 4-hour blocks, there's 1, 2, 3. So if you pick 2 of those and attend them, that's how you get the points
Do
for
you
it.
show up here in this?
Yeah, but
I'll get to those little detail. I think we'll probably show up at Dr. Flint's farm.
Okay,
Yeah, and I'll have maps and everything.
okay.
And honestly, sir, you want to bring the kids?
But let's say we.
You might not want to, but if you do want to, like here, I'm thinking at the end of this, block here, we're gonna have tanks set up by the creek with the fishes that we caught in them. And I'm gonna invite people to come and we can see, there's a lady coming, her name is, Ani Escobar Pop, and she's Georgia DNR biologist. She's like just a badass, cool person that I'm never got to meet, hang out, so, yeah,
Is it just with nets, no rods for the fish?
I'm sorry.
Are you just fishing with nets, no rods?
Yeah, none of the free fishing. Well, actually though, I'll tell you, a lot of people go fishing their whole life and they don't ever see these little ones. It's these little fish like the, darters and shiners and things that are really beautiful colors and stuff. So, no, that said, Friday night, if people want to come and there's some really nice big bullheaded things in the lake, we can go and do some fish and stuff like that. Yeah, out.
Can we bring, like, friends?
Yeah.
Okay.
Absolutely. People have shown up intoxicated. That's not cool. I mean, I appreciate the enthusiasm.
You
The
said-
dedication.
Wait, you said bonfire, I'm just saying.
Oh, yeah, speaking of which, if you have a
It's
bonfire,
not equivalent.
bonfire
It is possible.
there, I'm
I've-
very asthmatic.
You don't have to go to the bonfire. It's no
Okay, good,
problem.
because I'm very asthmatic.
We're good. No, it is possible, Sarah, to attend a bonfire with no intoxication. I've tried it. It works.
There.
Yeah,
It doesn't actually burn the alcohol in the blood. Okay, so here's this, tardigrade pictures assignment. Okay, and I guess the big picture is I have a box of slides. For 3 slides, I want you to take the slide, put it on a microscope, take pictures of that slide. save them to the computer, that's what you're gonna do, okay? Tardigrades,
Can I ask why it's called that?
I'm sorry?
Can I ask why they're called tardigrade?
Oh, it has to do with the way they walk. They kind of amble, and the first person that saw them in there, so like plantigrade is a specific way of walking, tardigrade, and I'm not sure what that.
I'm guessing they walk so slow they'll be tardy.
Yeah, it could be. I don't know. There may be an etymological connection there. I'm not sure. But I'm telling you, these are really, actually, one of the boblets is we had this guy, and I linked to him here. That was really a fun goblet. This guy, Paul Bartels, and he's one of the world's experts in, he's at Warren Wilson, and he, he's an expert in tardigrades. And I just, out of the blue, I was just emailed this dude. I was like, hey, I want to learn about tardigrades. So could you come to the boblets? Sure, he comes. It was so much fun. And actually, these slides that I have are a result of that boblets. I just have hundreds of tardigrades mounted on slides, which, you know, that's real cool and all, but I want to have pictures of them where I can actually do something with them. That's then documented. I can send them to a museum, and then there'll be documentation that we have that species on campus, right? So the point of this, why am I making you guys do this? Well, two reasons. One is I think it'll help you grade, because if you give me 30, 45 minutes, come to that little lab beside my office, and you do this assignment, you get some points, right? Number two, it's some transferable skill, right? Is that you are learning to use an inverted microscope, and you are learning to use a camera apparatus on the scope, which if you are doing histology, you're doing hematology, you're doing any manner of ecological work, being able to document things under a microscope is a useful skill, okay?
When does this due?
I'm just gonna make this due at the end. Right now, it's due, let me see. I can change it right here in front of your very eye. It's due on April 9th, but we'll make it, due April, what's the last day of class, is the 27th?
21st is the last day of class.
Really?
That's what it says on the syllabus.
Oh, sorry, it's not.
Okay.
It's the April 20. So here's the boblets weekend, then we have class on Tuesday, and that's it.
Wait, is that the exam? Like final exam?
The final supposedly is April 30th.
Yeah, right. So the last day of class is the 27th, but then the final is that next week. So this will be due the very last day of class. This, being the.
So the final's Friday from 11:15 to 2:15.
Correct. So where does it say the 21st is the, I must have just not changed that. Okay, but so here's some just, you know, the description of the assignment. So oh wait, actually I lied. For two specimens, two microscope slides, I want you to take these 3 pictures. So it's a total of 6 pictures. A picture of the whole organism. This is what a tardigrade looks like. They're always kind of the same. They have this is the mouth. This thing is called a bucopharyngeal apparatus. It's basically like a piercing, these pull and it shoots out this sharp little stylet and it punctures, and then they just suck up stuff. So this one's its anterior. This is the butt end, the posterior. And they have 8 sets of claws. 1, 2, 3, 4, 5, 6, 7, 8 sets of claws.
Because that would be the mouth, because it opens up like the alien does, the end, the right side.
Oh, like this? No. Yeah, this is the bucopharyngeal. That's the mouth end. So the things that become important when identifying them are this. So I want a close up of that, and the claws. So I want a close up of the claws, and then the whole organism.
Yeah, it looks like a baby million.
These things are real.
What's the purpose of the butt claws?
I have no idea.
No.
I'm telling you guys, I could just do a lecture on tardigrades. These things have this ability. They have a weird protein that no other organisms have that we know of, that when they desiccate, they go into this thing called a ton. All the water leaves their body and they just become this, like a little cyst, like a bacterial cyst. And then once they rehydrate, they just come back to life. But the amazing thing is when they're dehydrated like that, they have put them in the vacuum of space. They put them nearly at 0 degrees Kelvin. They put them at like 300 degrees Fahrenheit. They have irradiated them, and then they put water on them, they come back to life. They pulled moss samples out of a freezer that had been in the freezer for 30 years from Antarctica. And in that moss, frozen for 30 years, was a little tardigrade. And it just came back to life and just started laying eggs and everything. Right? Very cool animals. So you're gonna make two things like this, right? 2 different specimens, whole organism, bucopharyngeal, and claws.
Do
Okay?
we need a certain type of camera or?
It's all, this is this is the camera that you're gonna use. I hate to hear my own voice. So this is the little lab outside of my office. And this video should give you everything you need to be able to go into that lab, sit down at the computer / microscope / camera, and do everything you need to. Okay? Piece of cake. Should be interesting to do. So I think what I'm gonna do is I'm just gonna set up, I think that it's just too open-ended. So what I'm gonna do is on our Bio 280 Canvas course, I'm gonna make appointments on this calendar. And you guys can sign up for some appointment and go in and do it at that time. I may or may not be there, right? But this sounds stupid because it's like, why do you need an appointment? But then I just feel like if you make yourself an appointment, you're gonna go at that time, right? And then you'll know there's not gonna be somebody else in there. And I encourage you, this is something you can do this week and get it done. Right? A lot of this stuff is stuff you can get done now, if you've got the time, okay? So that's so that's the tardigrade pictures. Basically, make an appointment on the Canvas. Okay, the lab report, I've got the description written. I need to make this video, it'll take me no time. I'm gonna put the file with the information on the logs. Basically, this is an Excel file, which you'll fill in, based on the videos, you'll fill in the number of animals. And you'll basically then use the video and how to run the statistics in Excel, and just use this Excel file to calculate your statistics, and then you'll write up your lab report. Okay. My goal is to have all that up tomorrow.
Wait, have all this up tomorrow, you said?
Yeah, have all this stuff up there, i.e. the ball's in your court, right? Give you everything you need to be successful on this lab report. Our discussion last week is recorded, and that video is gonna be up there with this too. Right.
Video on what?
So there's gonna be, these videos here are the actual ones of the animals. Yeah, these are the animals crossings. So you'll count those, you'll see if there's a correlation with the log crossing score on this file, and then you'll write up your lab report. Okay. Intro methods, results, discussion. Okay, so I think, I hope my goal is for you to feel a little bit less anxious about everything. I feel good about where we're at this semester. I feel good, like we've been proactive, we've been pushing hard on the tests and things. but that will benefit us in the end. The boblet shouldn't be something that you're worried about. It should be something you're looking forward to and excited about. The tardigrade thing, I'm telling you guys, it'll take you 30 minutes. Maybe a little bit more, just depending on how comfortable you are with a microscope. The lab report is a Bearcat. This is a pretty big assignment. But break it into blocks and start on this part of it, and anything that I can do to help you. Yeah,
So is everyone in the class gonna get like 2 different ones? You have that right? Tardigrades?
yes, yep. It outlines in here, there's a box with, to be photographed, and then you'll take yours out and you'll log it in and put it in a photographed box. So that in the end, I should get pictures of most of those specimens. So there is an ulterior motive too, which is I want those pictures. Right. But then I can put them up like on iNaturalist and people can identify them, and I try to design things so we're kind of making some progress. Wouldn't it be nice to know like every animal that lives on a campus, right? That'd be cool to have documented. That's the idea of the bobbleheads.
And lab reports due the 27th as well?
Yeah,
Okay.
yeah. And, I go back and forth on the philosophy behind due dates. I feel like if I just put everything off to the end, that's gonna screw some people over because they're just gonna wait till the 26th. But, that's a lesson we all have to learn in life, right? I don't wanna make it too early either, so. Okay, good talk. Thank you for those. It was my intention to talk about them, but let's see.
So
Yeah,
you said we get graded for, well, I have 2 questions. One for attendance.
attendance does not function at all in this class.
Oh, okay, good. Okay, so I thought, and then, at Frankfurt. Oh, test rate, or test 3, or test 2, when would that be in?
Somebody just finished taking it this morning, so,
Gotcha.
yep. Again, with makeup labs, it's just seemingly out of control, I feel like, but, so therefore, sometime in the near future.
And when is, test 4 going to be due? Like,
Just check on the syllabus. Okay. So, last week, the online lectures were about the chordate phylum Chordata. So that is kingdom, animal, phylum Chordata. And then there's several subphylum. There's the Euro Chordata, there's subphylum Cephala Chordata, and then subphylum Vertebrata. Okay, we finally arrived at the animals that some people thought we were going to talk about all semester. All the animals, if I asked you on the first day of class, your favorite animal, they're probably all in this subphylum, right? But we can't ignore all the rest of the animals, okay? And that we started breaking that out into here's the big, in terms of the diversity and the taxonomy that you need to know for this class. This is a key slide, okay? You should definitely know these are all in the subphylum Vertebrata. We have superclass and then these classes, okay? Should know where these fall on, particularly this clay ground, right? Whereas the agnathans, the early stages of chordates, these are vertebrates. In fact, the hagfish is sort of like a barely vertebrate. They don't even have vertebrae, right? But they don't have jaws, they don't have teeth, they don't have a lot of things. Right? But we're going to include them in this subphylum Vertebrata. Okay? So then we get the vertebrate, they do have a vertebrae now, from here up, the lampreys, but they don't have jaws. Right, jaws occur here, right? And then we have the cartilaginous fishes, sharks, skates, and rays. That was the second lecture last week. Or no, we didn't even get to that, did we?
No, we didn't.
And that's we're gonna pick up here today. And then we're gonna get up to this group, the Sarcopterygians. Sarco means like fleshy, so this is the fleshy fin fish. And then the rest of the semester, we're just talking about this little branch here, the tetrapods, includes amphibians, reptiles, birds, and mammals. Okay. We talked about, that's what we ended up talking about jaws and the importance of jaws and where they came from. So certainly the first jawed vertebrate would have looked, would have been a cartilaginous fish. Bones came a little bit later in evolution. But we're talking very, very old group of vertebrates here. Okay. Not to say that something like a great white shark is primitive, right? But that lineage that includes the sharks, skates, and rays has been around a very, very long time. Okay. So they're kind of this weird mix of ancestral and derived, right? If you look at a goblin shark, do you ever see goblin sharks and their whole jaws are detachable and they can come almost, that's very, very derived. But yet, their blood content, their osmotic balance is very ancestral. Their kidney doesn't really try to work on maintaining a gradient between their blood and the seawater. Their blood has just as much stuff in it as seawater. Right? They don't have bones. They just have made work with cartilage. Right? So there's this weird mix of things. This class can be broken down into the holocephali, and the elasmobranchs. Okay, holocephali. This is a pretty minor group of these really strange fishes called rabbit fishes, rat fishes, chimeras. Most of the chondrichthyes, 810 out of whatever, are in the elasmobranchs, including sharks, skates, and rays. Okay, really incredible diversity in this group. The sharks, skates, and rays.
That's cool.
Yeah. From this perspective, I like this overhead.
Dun, 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 dun.
This is incredible diversity. If you guys have not gone to the Georgia Aquarium, you've got to go and be in this walkway and see 3 different whale sharks swimming over top of the largest fish in the ocean.
They have a sharks exhibit now too.
Mm-hmm.
It's pretty cool.
Yeah, that is pretty cool.
That
They have what?
A whole shark exhibit. Yeah, they have, I think it's 3 whale sharks now.
Yeah, but they have like,
We're up to the
what do they have,
4th one.
lemon, nurse sharks
They have
and,
nurse sharks, they have the,
bull,
leopard shark.
yeah, maybe bull sharks or something.
What's that black one?
Wait, I thought they had 4 whale sharks.
This is that one that lives
They
deep,
went dive and stuff.
it's a really enigmatic species.
What are they doing?
Is it maybe megamouth shark?
What?
This
It's
may
really
be,
hard to get more whale sharks.
Is it a Greenland sleeper?
I'm thinking that maybe it's that megamouth shark. I'm not sure, I can't read it.
How
So,
do they get whale sharks in the Georgia Aquarium?
oh no, that's the megamouth.
They have a whole display on it.
Yeah, that's.
That's the
Yeah.
mega
I
mouth.
think it is the megamouth.
Yeah.
Yeah, so, I mean, what can you say about sharks, right? These things are so darn cool. From little species, the banjo fish, or this is a sawfish, which is in this group of fishes, to some of the biggest predators on Earth.
Those sawfish are
Um,
huge. They have one at
Very
Ripley's
cool. So you
Aquarium.
see this variability in the size, morphology, ecology, some things that unite them though, right? The cartilage, so that their skeleton is not ossified, it doesn't have those. The minerals deposited that make it heavier and stronger, right? That's bone. But cartilage is much lighter weight and very flexible. The scales, the type of scale we find on sharks is called a placoid scale. And they're basically, the thought is that the first tooth is a derived version of a placoid scale. Right? So remember when the first jaws evolved, they certainly wouldn't have had teeth. Jaws became before teeth, but they would have had skin covering them, and the skin had these placoid scales. And so the genes for the placoid scale just stay on longer and make those scales really big, and that becomes a tooth eventually. So the pre-existing placoid scale becomes a tooth, right? They even have a pulp cavity they have. So if we look at, here's those placoid scales. They're heavy and they're very almost bony, right? They have dentine and enamel, right? Covered.
So they have some tooth matters now.
Yeah.
They're also called denticles, the scales are, and
Oh.
because of the dentine in them.
Yeah. So there's that tooth connection again.
Yeah.
Very interesting. Most fish, when we think of, have these cycloid or tenoid scales. They're very lightweight, flexible. They just kind of come right up. You can actually make like nail files with shark skin because it's sort of like exfoliating your skin. So that's a thing that is the Elasmo bring to share. Reproduction. This is a kind of another way of grouping animals, or another thing I'm going to be looking from here on out, looking for about the different groups is whether the sperm and the egg come together outside of the body, that's external fertilization, or whether the gametes, whether fertilization happens inside of the body. That would be internal fertilization. Okay. And we can talk about the implications of that, but right, if you think about this basically means sharks are internal fertilization. There's some way of transferring the sperm into, in this case, in the female's body, so that it meets the egg inside of her body. Right? And we'll talk about what that is. We saw this firsthand with our little dogfish shark, right? What, she had two little babies, right?
Was she had two?
Yep. And sharks don't lay hundreds of eggs that then hatch into hundreds of little baby fish. They often retain the eggs in there and just have a very few offspring, right? But they do put a lot of effort into those few offspring having high survival rate. Right? So it's just a different way to achieve the same thing. You either have lots and lots of babies. I have a friend, she has 6 kids, and I mean, it's just kind of like some of them are probably going to make it to adulthood, right? And then we have a couple kids, and we just like, if they need their butt wipes, then we're right there for them. It's like 2 different ends of the spectrum, right? And sharks are at this, have a few and put a lot of effort into them, right? They have big eggs. The eggs have a lot of yolk, okay?
So the babies come out larger, which increases their
Yes.
chances of survival.
Exactly right. Yep. Yeah. And so if you think about it, it's just a different strategy. The problem with it is when shark populations go down, it takes a lot longer for them to rebound, right? Because it just, with the few offspring thing, the populations don't grow very quickly. Another really important terms here. Paris means life. Ovi means egg. So these are egg-laying species. So most fishes are oviparous. Most amphibians are oviparous. We only have one species of oviparous mammal. Or No, actually, I think there's three, all the monitoring. But, and some sharks lay eggs. Other sharks have what we call ovoviparous. Okay? So here's the gradient. Egg layers, live bearers, viviparous, live birth. So here, the baby is laid not in an egg. It's basically given as live birth. And the other thing that separates these two is in the viviparous species, like humans, there's a connection between the mother and the offspring in her body. Okay? So the babies, while they're growing inside of the mama, have some nutrition that's provided directly from the mother to the babies. Okay, here in ovoviviparous species, basically the eggs are retained and they hatch inside, so that when they come out of the mother's body, they're alive and not in the egg. Okay, but there's no connection between the mother and the offspring.
So viviparous is like the umbilical cord.
Yes, right. There's other ways
So
to
is
do it.
that round thing that was connected to the shark baby?
That would be a classic example of why ovoviviparity is that the yolk is providing the nutrients.
Okay.
So there's no direct connection. So in other words, another way to think of it is mama pregnant shark can drink alcohol and smoke cigarettes. It's all cool, right? There's no fetal alcohol syndrome in sharks.
Like a kangaroo, basically. Like, except it's kangaroo, but it's on the inside. It's like a little
Yeah,
pouch on the
kangaroos
inside.
they're kind of a weird example because they do have this, but their placenta, that connection between the mom and the baby is really short-lived. So there is a connection at some point,
Okay.
but then that little joey detaches, comes out of the womb, crawls in, latches onto a teat. Well, I guess it's not teat because they're in their pouch.
Yeah.
So you think of, here they lay eggs quickly, here they give live birth, here they just keep the eggs inside of them until they hatch. That's what I think about it. And therefore there's no connection.
So the two sharks that we found, were they had already been like,
They probably would be ovoviviparous dogfishes. So in other words, she's gonna keep those little babies grow inside of her, eating the yolk, and then they basically emerge from the membrane, and then she lays them. Or a lot of times, the mom lays the egg, and then immediately this little baby shark just comes out of this little thin membrane, right?
and they go back inside or to go back into a, like,
Then they're just ready to rock and roll, you know, to do like baby shark stuff, I guess. Maybe that would be. So this would be an oviparous species. We'd see, have you found these at the beach?
Mermaid
Skate, yes, they're called mermaids first, they're usually like skates or ray egg
I've seen
pouches.
them on display at aquariums.
Yeah, so here's one, and you can see the little, here's the yolk inside of it.
that's sharks.
Mhm. Some types of sharks have mermaid purses. Yep, pretty cool. Lateral line system is a way for an organism to monitor water pressure. Okay, so if there's a fish swimming by them and it moves the water, then that pressure difference can be received and interpreted by the lateral line system. So fish can tell differences in water pressure. They have these, sharks have these things called ampullae of Lorenzini, which detect minute differences in electrical impulses. All right. So what in the ocean is giving off electrical impulses that sharks would want to detect? Why is a shark, have they been adapted to detect very minute electrical impulses? What are those electrical impulses?
Some be like dolphins and
It could be
mammals.
a heart rate of the fish.
could come any other living thing, basically. The nervous system is based on voltage differences. And so actually, yeah, so it may be predatory thing or defense of dolphins, but a lot of times it's, they use them to detect their prey down inside of the, maybe inside the substrate. And then chemicals, chemoreception. Hammerhead is just a surface area thing, right? It's a way to increase surface area for chemoreceptor cells, probably some electroreception too. Of course, they can see, but it appears that their vision works better in low light. So they are sort of adapted to low light conditions. So you see these little pores here, these ampullae are little pores. They're fluid filled and they have these cells that are specific.
Is that a tiger shark or anything?
Could be,
Sorry, just looking at the shape of the snout.
yeah, could be. I'm not really sure. Some really neat adaptations here with shark and the way that they feed. And this goes back to, again, the form and function. The structure of their skull is very unique in that the jaws are not hard attached to their skull, right? Like our jaws, our upper jaws melded and just fused with our cranium, right? With sharks, they're two separate units, right? The jaws are actually suspended from the cranium by these ligaments. So that allows the jaws to protrude out of their mouth, basically. So you can get a huge gape. The mouth is open a lot wider. And so you'll see these pictures of sharks. Here's those gill arches. There's the vertebrae.
So the gills are actually part of the vertebrae.
No, not necessarily. Here's the gill arches, and then there's the vertebrae. They're just down below it, and these are gill slits. And so we see this. This used to be one of these, right? The jaws, but here's the jaw too. I think this is the mandibula and the palatal quadrate or something. And rather than being fused with the skull, they are movable, protrudable, which allows them to protrude the jaw out of the mouth. You hear reports of sharks eating things like dead whales, and you can see these huge chunks of the flesh that have been taken out by, again, the ability to move their jaws outside of their mouth.
So why can they eat dead prey? Like most animals don't eat dead prey, but sharks do.
Say it again?
Most animals don't eat dead prey.
Oh, okay.
So sharks do. Why is that?
I mean, I would say that most animals do eat that prey. Most carnivores do.
It has a,
I was surprised that a lot of times if I shoot a deer and I get the gut pile, then I'll put a camera on it. It's amazing. Coyotes come to it. Owls come in eating from it. Hawks.
Well, like birds, I can see that. Like mammals,
Yeah.
most of them don't. They stick to life prey.
well, they have
Not well
to
injured
it
prey,
somehow.
but like
Yeah.
dead animals.
I guess it's probably just a lot of bacterial
Yeah.
issues there.
But like I'm saying, it's like it does not affect the shark because that's why most animals don't eat dead prey because they get sick. I mean, they probably put two and two together. That when I eat this dead prey, I get sick. But like sharks have a strong immune system.
I guess. I don't know. It's a question.
Oh, when they eat a seal, it's dead.
I'm not really sure.
Well, it dies instantly. Like it's dead, and then it's
Right,
dead for a
doesn't-
while, and then it goes and eats.
It doesn't have time to
It's
become
fresh.
like super infected.
Yeah, it's
Yeah.
fresh, true, it's fresh meat.
Oh,
Ooh.
oh,
Yeah, that's a problem.
that's
That's a
bad.
problem.
Yeah, that's not kind of a problem.
Walk it off.
You're not gonna just lose your leg there.
Miss Fencing, the child is injured on the playground. I think think he's probably okay though.
I thought you were gonna call me that.
Little Johnny, minor questions on the slide. It's funny, not
He
funny.
hit him pull really hard and his leg exploded.
I'm groovy. Okay, so, class actinopterygi. Think of this PTER, this is another root word that you're gonna see quite a bit, PTER. This is kind of like limb or wing, like pterodon, pterodactyl, actinopterygi, sarcopterygian. When I see this, I think of a limb, whether it's a wing or whether it's a
Wing, yeah.
fin on a fish. These are the ray fin fishes. There's about 60,000 species of vertebrates, and 24 to about 30,000 are in this group. This is a very diverse group. Most vertebrates are in this group right here, okay? And this includes pretty much all the fishes that you think of. These are bony fishes, not the cartilaginous fishes, right? Their scales are not those placoid scales. Their fins have these little parallel radial bones. So if you want to draw a actinopterygian fish, here's the fin, and they have these parallel. That's the ray fin that comes from those little rays in their radial bones in their fins. Right. We're going to talk about the sarcopterygians, and then you'll see the comparison here. And I don't think I'm going to go any deeper in the class actinopterygian. Anybody know what kind of fish this is?
Sturgeon.
Yeah, these are there's not too many species of sturgeon that are in good shape. But I think we have an Atlantic sturgeon here maybe. How about this one? These are very primitive type of actinopterygia. These are big, say again?
Cigar?
Is that alligator?
No, that's not.
No, not a
No,
gar.
that's not gar.
They're sort of like filter feeders. They open their mouth huge like this and just troll through the water. Look at the gill, look at how big the gill opening is relative to the body. This is a paddlefish.
Yeah,
They live in
that's
the Ohio and Tennessee River drainages. Of so many fishes, squaw fish, all these different shiners. There's probably like a buffalo or a drum, suckers, catfishes. So many different. This is what Keith studied for his master's, these armored catfish in the southern
Catfish.
Oh, I recognize that one
Sunfish.
over there. It's a red-tailed catfish.
That's a red-tailed catfish.
Are those catfish or
Those get
suckerfish?
huge.
These are all
Yeah.
catfish.
I got those with the Ripley's.
The little
They're
barbels.
huge.
But they have the subterminal mouth.
Yeah.
And so, talk to Keith about it sometime, if you got like 4 or 5 hours. Salmon, trouts, there's a grayling, all the different, I mean, just beautiful, they're all in this group. And we're not even talking about all the ones in the ocean, right?
Those are, I always like those. Those are always.
Oh, I've caught one of those
Rockfish.
before.
Rockfishes, you caught them? Some of these
No,
are,
I caught one by mistake.
some of them are venomous, right?
Yeah,
No,
they're very.
Yeah, I caught him in, I was just like shaking it off trying to get it because I was afraid it would get, stung. I think I killed it.
okay, and then, so that brings us to the sarcopterygi. Sarco means sort of like, I think of like the, sarcophagus. I don't know other roots with that, but I think of like fleshy. These are the lobe-finned fishes. And so check out this part of the limbs of the fins, right? The fins are attached to the body with one upper arm bone, right?
Yeah, wouldn't they be very easy to remove?
The fins? Yeah, I guess so. But they can also exert a lot more pressure on their fin with that attachment.
Oh.
So this is a homologous. That bone is homologous to our humerus, right? It is attached just like ours are. This is the the lineage, right? So if we look at it like this, then we have our, here's the deuterostomes coming over here. We have the echinoderms. We have the chordates, include the urochordates, cephalochordates. We have all the hagfishes and lampreys. We have the chondrichthyes. Here's all the bony fishes, 24,000 species. The sarcopterygians. This lineage leads to us. It leads to the amphibs. It leads to the reptiles, all the tetrapods. So this is not to say, this is a coelacanth. This is not to say a coelacanth is a missing link or that it's our direct ancestor, but it would be on this lineage leading out to the mammals. Okay? They've been around for a long, there's really interesting story about coelacanth where they have fossils of these things from hundreds of millions of years ago. And they thought they were extinct many, many years ago. And then there's this person walking in a fish market in South Africa, and they're like, holy hell, there's a coelacanth, right? And it turns out that they are still extant, but they've been on this earth for a very, very long time. Okay? It's a weird group of fishes. The lungefishes, they live in very hot water with a little bit of oxygen. The waters dry up a lot of times, and basically they're adapted to being able to get around in shallow hot water. They have a lung. They come up and gulp air at the surface, right? So these are sort of like, it's intuitive that these would be the ancestors of those first fishes that made it onto land, okay?
Yeah, that's really cool.
Look at some, okay, so, oh wait, shoot. We'll talk, we're gonna revisit this group, okay? But I need to talk a couple more just sort of general topics of fishes. This should look familiar to you, right? It's just pelvic, ceca, the stomach, intestines, and all that stuff. Already, this group of vertebrates have all the same organs and organ systems that we do, okay? It's not a coincidence, right? That's to say that all of those things evolved back here and include the drum. In fact, they evolved back here, right? Include the dogfish. That's what we're doing in lab. They include the drum. They include the frog, which you're gonna look, some of you will see in class today, and they include us, right? So the presence of gallbladder, spleen, pancreas is a very, very old type of thing, right? So, here's another essay question coming up on this test. This is very interesting to think about how organisms regulate their osmotic balances, okay? So this is kind of like the logic, right? Is most of the body is made up of water, but the water also needs stuff. The blood needs things in it. It needs salts. It needs these ions, right? In order for the nervous system to work, we need calcium ions. But there needs to be a balance between these things. Okay? That's one of the sort of the aspects of life, is that cells maintain conditions inside of them that are different from the environment. That's homeostasis, right? So fishes have a really unique challenge in this regard, right? So these ancestral marine chordates, and so these would be like sharks and things. What's their approach to that? Is they basically maintain water ion concentrations very similar to the ocean water. Okay, so I've spent a lot of time in Japan, and in Japan, many years ago, the only fish they could eat in the mountains were sharks. Why? Because the meat has lots of urea, lots of salts, lots of things that make it preserve better. They could put it on a little cart and pull it all the way up to the mountains. And regular fish, that's not gonna happen. They're gonna be rotten very quickly because sharks have this kind of condition. So they don't bother to try to maintain much lower salt concentration than the ocean, right? They just kind of, it's more isotonic. Now, these two groups, derived or more recent marine fish and freshwater fishes, have this unique challenge because if you, and let me just show you the picture. Okay. Here's your derived marine fish, something like a grouper, right? Another way to think about this, and I don't know, some of you guys might not have done this experiment in 120, but you might have done this in high school. You ever done this where you have like a little, have a beaker, and then you take a membrane and you fill it with water, and you put the water-filled membrane in water here. So if you have fresh water, so i.e. no salt, and then you have salty water in this little pouch, what's going to happen? Now, I can tell you, water goes through the membrane. Water can go through this membrane. The salt can't go through the membrane. Okay, so if I take this pouch and I put it in fresh water, what's going to happen to the pouch?
The water's going to go into the pouch, it's going to expand the salt bag.
So the water,
Will go into the salt bag
why?
because it will go to the high concentration and it will.
It goes from an area of high concentration. So if you think about it, here, the concentration of water
Is much higher.
Here, the concentration of water is low. Why? Because there's salt.
And there's low salt on the outside, but high salt on the inside, so.
The water-
flip-flop.
That's right, so water will tend to increase, keep going into the bag, and it's going to expand. Okay? Here's the other analogy. Is a blood cell in your blood, and you drink 6 gallons of water really quickly. What happens? Then the water can, you've heard about this, people drink so much water that they die.
Yeah, the blood
Does it go to the
burst.
hospital? Same kind of thing. This is osmosis. This is what we're talking about here, right? This is what for like a freshwater fish, that's the challenge. It has some salt in its blood. It needs salt for the nervous system and so on and so forth, right? So in this case, there's a lot of water outside relative to what's inside, so that fish is constantly taking on water, okay? And it has to get rid of the water. And it needs to maintain and keep those salts. The salts are precious. It needs to maintain a higher concentration of salt in the body than in the water. So how does it do this? It excretes dilute urine. So the kidneys are making lots and lots of urine, pushing out the water, okay? It keeps the salt because the gills are actively absorbing salt from the water, right? So it's actively transporting salt into its body through the gills, and it's actively flushing out the water through its kidneys, okay? The kidneys have resorption of the salt, okay? So it's all about keeping the salt and getting rid of the water. Now, what do you expect is going to be the challenge for this grouper? Where's the water concentration higher? In its body or in the salt water around it?
And the salt water around it, right?
It's body.
In its body.
Oh, yeah.
Because, and I don't know, this is again, my simple, like, 5-year-old version of understanding is, if there's salt there, there's no water, not as much water. The water sort of takes up some space that water could be. The salt does. So if you have salty water, it's not as much water there. So here, the challenge is to get rid of salt, right? And to take on water and keep that water. So they drink seawater, the gills are secreting salt, right? The stomach absorbs some of the salt from the food, which is going to be voided with the waste, right? So here we're getting rid of salt by absorbing it through the stomach and then pooping it out. We're getting rid of salt by secreting it from the gills. And then what do you suppose, what's this going to say about the urine of this fish? Is it going to be concentrate or is it going to be dilute?
Very diluted.
Concentrated.
Okay, why concentrate, why dilute?
Well, if it has a high salt content on the inside, we wouldn't want to keep the salt content, so keep it diluted.
Well,
But it would be backwards though, because it has a high salt concentration.
they have too much salt,
Oh.
right? And what do they want to, because if they're keeping too much salt in their body, then they're going to be out of osmotic balance. So they want to get rid of the salt and keep the water. So therefore, just like here, we're dilute urine, getting rid of a lot of water, here is excretion of little water, so very concentrated urine, right? So there's a, check out, there's several explanations of this on the web. Spend some time with this diagram, particularly looking at, what the function of those different organs are and what the challenges are for those different kind of fish. Then there are some fishes that spend time in both, and I guess they must have to reverse this. Say like a salmon that swims up into the freshwater river, their gills must switch when they go into one or the other. Right, challenge of getting oxygen is the water has about like 1/20 of the concentration of oxygen in the atmosphere. So gills are very, very efficient at extracting oxygen from the water. Right, so this is countercurrent exchange. The water flows over. This blood is arterial blood, which is coming from the heart. It doesn't have oxygen. So oxygen diffuses into the water, or from the water into the blood here, and then goes out to the body, right? This blood is coming from the venual system. It has a lot of carbon dioxide, and it diffuses into the water and leaves the body here, okay? This comes down to what's going on in the mitochondria. Mitochondria need oxygen. It's absorbed here. They're producing CO2. It is diffused into the water here. Buoyancy. We saw on the drum that we dissected above most of the viscera, but below the kidney, right? We see that big balloon-shaped organ, that's the swim bladder, that is in these actinopterygiae, and it is there. Is really highly vascularized, so we've got a lot of blood flow. The oxygen from the blood can leave the blood and go into the swim bladder and expand or contract, depending on where the fish needs to be in the water column. That saves the fish a lot of energy because they can just sit there and maintain their place in the water column without having to swim or use their fins or things like that. So that's another adaptation of these fishes. Conservation, sharks, this is a big story right now. People are harvesting shark and throwing 98% of the biomass back into the ocean, right? They catch a fish, they cut the fins off, maybe a couple of steaks, throw the rest of the fish back, right? Takes way too many shark fins to satisfy certain parts of the world in their hunger for shark fins. The problem we've talked about, these are long-lived fish, they reproduce very slow, right? And so we see huge declines in that there's declines because the catch that they're bringing to the shore just decreases every year. It's not a sustainable. Sure.
Why did they go for the fins?
It's probably just in some legend or something. It's all cultural. It's nothing to do, I don't think to do with taste or, I have no idea, honestly. Not to be,
I thought it
Yeah,
was like
because I know shark teeth, I know like people likes to buy those, but I know aquariums can easily go to the bottom of a tank and grab, shark teeth real easily.
oh, the teeth, yeah,
Yeah, because sharks shed so many of their teeth.
right, yeah,
Aquarium can easily go down and grab shark
Yeah.
teeth and sell those real easily.
that's true. So here are some there's some characteristics of fishes that are going to become endangered. If you're a fish that is big, like a tuna, if you're a fish that takes a long time to mature, lays very few eggs, they are very top predators. They live in dense congregations, you're screwed, right? This is like basically, we're talking about tunas right here. Tunas are so valuable that they fly helicopters and shoot them with harpoons, right? And they can sell them for tens of thousands of dollars, right? And so the other thing is, if you have live in just a small part of the earth and you specialize in a really specific type of habitat or eat a specific food or breed, some of these things breed only like laying eggs in the underside of a log in a little hole or lay eggs in a certain size of cobble in the creek. These are all things that are going to make you susceptible to becoming in decline. We're going to see these on the bollets. This is Ephios moscata, the Etowah darter. And we have some really neat fishes here that are protected. These are on the U.S. Fish and Wildlife Service threatened and endangered list. The Etowah River is a hot spot for fish diversity. And I think that's it. Okay. Whew, that was a lot. I appreciate you guys' attention and keep talking to me about this, all these, particularly that laboratory stuff. I need to get that stuff to you guys. So I always appreciate reminders and things like that. Don't be bashful about coming to me and getting what you deserve. So.